Bon dia. I will introduce Luis Leria. Sorry if I kill your last name. He has a pharmacy degree and a master degree, PhD degree that he obtained at the University of Campinas. He did a postdoc at the Joslin Diabetes Centers in Harvard. And he got a fellowship for this postdoc. And now Luis is an assistant professor at the Department of Pharmacology and Cell Biology in Ribeirão Preto. He is a principal investigator at the Center for Research on Inflammatory Disease, and he is an affiliate member of the Brazilian Academy of Science. And his interest in study cell-cell and ligand receptor interaction within the thermogenic fat and white adipose tissue niches. So Luis, welcome to this workshop. I will show you when you will still have 5 minutes left. And the term.
Thank you. Good morning, everyone. First, I would like to thank the organization, especially to Adesio, for the invitation. It's a great pleasure to be here in the medical school, which is a very fancy venue. Love this place. So it's a great pleasure to be here. So I will present today for you this ongoing study in our lab, which was actually has been brilliantly conducted by a PhD student, and all of the data I will show for you today is unpublished data. So I just would like to kindly ask you to not take pictures of the slides with data. Okay, the others I think are not that interesting, but it's still, if you want to take pictures, it's fine. So I think one of the major things like to introduce to you is that adipose tissue is not all bad. We have different kinds of adipose tissue and adipocytes. So of course the one we associate more with fat is the white fat. So is the fat that has this unique morphology. It is unilocular and has this what we call the eccentric nucleus, which is like a nucleus that is in the periphery of the cell, and this cell has a big, only one lipid droplet, and it's make it specialized in store fat, store energy in the form of fat, form of triglycerides. And the expansion of this tissue is associated with higher risk for type 2 diabetes and cardiovascular disease. But on the other hand, there is this very interesting type of fat that we are more dedicated to study in our lab, which is the brown fat. The brown fat is composed mainly by, like, the whole parenchymal cells are the brown adipocytes, which are multilocular. They have a multilocular morphology, completely different of the white fat, and this is more specialized in consume the macronutrients from the circulation, like fatty acids, glucose, branched chain amino acids, in order to generate the heat, right? And more brown fat you have, lower your risk to develop type 2 diabetes and cardiovascular disease. And the beige fat is an intermediate kind of fat, which is an inducible kind of fat, and consists in the surge of clusters of brown like adipocytes, which we call the beige adipocytes. They are brown-like because it's not the same, actually. They have a different genome profile. And it's induced by cold, and the expansion of this beige fat is also associated with a better metabolic phenotype, with a lower risk for metabolic and cardiovascular disease. And historically, brown fat has been associated with the newborns, like in humans at least, or in hibernating animals in nature. But in humans, it was traditionally associated with the newborns. So they have plenty of brown fat in a phase of their life where you have very low muscle mass. But this has been changing over the recent years. And in 2009, there were some interesting back-to-back papers published showing the importance of this in this tissue in adult humans. And even more recently, it was shown that in this very nice paper published in 2021, in which there were 10,000 people screened in PET CTs, and they were divided in those patients with detectable brown fat and those with undetectable brown fat. And as you can see here, the ones with detectable brown fat in the brown bars, they have lower likelihood to develop type 2 diabetes, dyslipidemia, and cardiovascular disease. And moreover, you can see here the glucose levels of them. The ones with positive brown fat, they have lower fasting glucose levels. And this is even more evident in those with higher BMI. The same happens for triglycerides. So basically, this means that when you have a more active brown adipose tissue mass, you will clear better the energetic substrates that are circulating, okay? So this causes the protection against the disease. And brown fat is very richly innervated, and its function is very fine-tuned. Is fine-tuned by the integration of these efferent and afferent signals. So basically, the main innervation that goes to the brown fat is arise from the efferent ones, arise from the hypothalamus, and is majority noradrenergic, but can release not only norepinephrine, but also NPY and also ATP. This ATP can be converted in adenosine, and adenosine can also regulate brown fat. But there is this very way less explored innervation in the field that is the sensory neurons. These sensory neurons, they can detect signals here in the brown fat, like they have some metabolic receptors also, but they also have mechanosensitive receptors, and they can detect changes in adipocyte function and signal to the dorsal root ganglia, and this can convey this signal to the central nervous system to regulate the efferents. So if the thermogenic activity is low, it can be perhaps suppressed, and then you have a higher sympathetic outflow. But there is also another function by the peptidergic ones that release peptides here that are very important for the tissue function too. So basically, these neurons, they have this they are connected by this dorsal root ganglia, and you have the nerval ends that goes to the central nervous system, but also the end that will release the neuropeptide and also sense the change here in the tissue. So it can propagate the signal to the central nervous system, as I said, but also you have the release of neuropeptides that can regulate tissue function. And the sensory nerves that innervate brown fat and white fat, they are different. Their dorsal root ganglia are located in different parts of the medulla, in the spinal cord. And the ones going to the brown adipose tissue are mainly in the cervical 4 to thoracic 2, while the ones in white adipose tissue are thoracolumbar ones. Okay? So they are different, and the nerves are also can be different. These sensory nerves, they are way abundant in the skin, for example, with the function to protect us against like harmful stimuli, like the heat or mechanical stimuli. For example, these piezo2 fibers, they are mechanosensitive and chemical sensitive fibers. There are others that are the C fibers that are the ones we are focused here. And the C fibers are very nociceptive fibers that are sensitive to heat and sensitive to capsaicin. And they are very important in the protection against heat stimuli. Okay, and then these sensory nerves, the nociceptive ones, they can be peptidergic or non-peptidergic. The non-peptidergic are the ones that I said in the beginning that are the ones that can convey this the stimuli to the central nervous system. But on the other hand, there are the peptidergic ones that release especially CGRP, substance P, and neurokinin A in the tissues. And recently, there were those two studies published back-to-back in Cell Metabolism, just showing that the mechanosensitive nerves, which are piezo2 positive nerves, and piezo2 is the receptor in the nerve ending into more close to the tissue. These sensory nerves, they actually are stimulated to to suppress the sympathetic output. So basically, when they are active, normally you have lower sympathetic tone. But when you suppress piezo2, you have an increased norepinephrine release in the in the fat and producing browning, or can produce the activation of even the brown fat. So basically, they have they have elucidated the role of piezo2 positive fibers in the regulation of thermogenesis. But regarding the nociceptive ones, we know too little about. And in the 90s, there are some studies showing that there are indeed some positive substance P and CGRP positive fibers in the brown fat. And here, as you can see, those CGRP positive nerves traveling in the brown fat. Also, other studies, they denervated this with capsaicin. You can add if you add a high dosage of capsaicin, you can deplete these nerves, and then they could see reduced response to cold in terms of UCP1 levels when you use capsaicin. But capsaicin also reduced the amount of beige cells into the periovarian brown adipose tissue in female rats. So there are indications that indeed this nociceptive innervation do the opposite of the mechanosensitive ones, because, as I just said, mechanosensitive neurons, they negatively regulates the sympathetic tone in the tissue. And it appears here that this is actually positively regulating this sympathetic innervation, or perhaps independent of sympathetic innervation, because when you take it out, you don't see, you actually lose thermogenic capacity. So this is very actually not really well studied, and that's why we decided to study more in depth this topic, and in the PhD of João Manuel. And as you know, most of the literature in this field is focused in the sympathetic nervous system, and more recently in piezo2 positive neurons. But we don't know yet how this nociceptive innervation is regulating brown fat, and in which process actually it affects it. It's regulating the adipocyte activity, or it is regulating the differentiation of de novo adipocyte formation there, or maybe it's regulating the immune system in the niche. So which process are these nerves are regulating? We don't know. So we decided to investigate more in depth. One of the first things we did was to make a retrograde staining with cholera toxin. So we injected cholera toxin in the brown fat, and then we look at the positive cells, CTB positive cells in the dorsal root ganglia in different regions in C5, in C7, T1, T2, and as you can see, there are plenty of neurons coming from actually connected with brown fat here. And as a positive control, we use the stellate ganglia, which is the main sympathetic ganglia that projects sympathetic nerves there. So, of course, if you inject cholera toxin in the brown fat, you realize that many of the nerves going there are in the stellate ganglia, but all of those are tyrosine hydroxylase positive ones. Okay, so this is positive control. But here's the interesting thing: you have sensory fibers coming from the different dorsal root ganglia to the brown fat. Okay. And here we just confirmed that those fibers are, we have plenty of nociceptive fibers there. Nociceptive fibers are positive for NaV1.8 and TRPV1, which are the main receptors. This is a sodium channel, and this is the TRPV1 is the one sensitive to capsaicin. And they have a huge overlapping. But here I put them in separate, so you can see that TCB overlays with NaV1.8, and TCB also overlays with TRPV1. On the other hand, we have a very few tyrosine hydroxylase positive fibers. We could find only one here, just to say that we didn't find any in the dorsal root ganglia. So in previous studies, actually from the Lier lab from Scripps and Adam Pataputian group, they found that there are indeed tyrosine hydroxylase positive cells in the ganglia that goes to subcutaneous fat. And here we are finding one or another that goes to brown fat, but there are really a few, and perhaps there are some special function that we still don't know. But it's just like an almost negligible amount. The first functional study experiment he did was to inject RTX. RTX is an agonist of TRPV1 that is 1,000 times more potent than capsaicin. And you can inject like 5 microliters of this to be restricted in just one lobe of the brown fat, while it doesn't leak to the other lobe, and in the other lobe you can inject the vehicle, okay? And we wait 4 weeks, because 4 weeks is the time to wait for these nerves to die, and then we evaluated this tissue, and as you can see, the side that got the RTX injection became more whitish. You can see a loose of the brown fat, typical brown fat morphology. Okay, so it completely changed and it starts to get some more unilocular brown adipocytes. And well, that was the first evidence that indeed these nociceptive nerves are regulating, positively regulating the brown fat function and perhaps the identity as well. And here, as you can see, the thermogenic problem genes, in comparing the lobe that received the RTX with the lobe that received the vehicle, you can see that there is a clear reduction in the expression of UCP1, PRDM16, SIDA, and which are very established brown fat markers, brown fat identity and function markers. And here's the confirmation at the protein level that indeed, when you denervate these nociceptive nerves, you lower UCP1 levels in a lobe-specific way here. RTX is quite aggressive, and as we know from recently papers from you at saying lab, there are some TRPV1 positive precursor cells that gives origin to very highly thermogenic brown adipocytes. So RTX is kind of dirty too because it can kill the cells, right? So what we did here was to target the NAV 1.8, we mated here NAV 1.8 Cree mice with DTR, inducible DTR flux. So basically we are conditionally expressing diphtheria toxin receptors only in adipocytes. Only in NAV 1.8 nerves. And then we inject the diphtheria toxin only in one lobe while we preserve the other. But when we did this, we could reproduce the data with RTX. So basically, the lobe that denervated displays a very white fat-like phenotype with a lot of unilocular cells, and they have reduced UCP1 expression at the RNA and protein levels. Here is paired, okay? So as you can see here, there is an important reduction in UCP1 levels. This, when you inject only in one side, in one lobe, functionally, the mouse can still defend their body temperature in cold. They are fine. But then we injected in both lobes to see how it can impact the thermogenesis, the thermogenic machinery, and the adaptation to cold in these mice. So we injected diphtheria toxin in both sides, and when we did this, you can realize that the animals go hypothermic. Okay, so this is in cold. This is a cold challenge. We put them in cold and measure their body temperatures every hour, and as you can see here, their body temperature goes really, really down. Then we did another strategy to avoid here is to clear even more. Like we injected intrathecal the RTX just to get the sensory fibers coming from DRGs, because eventually there are some that can arise from stellate ganglia, which is another work we are doing too. But here we restricted the RTX just to the spinal cord to reach those ganglion, the dorsal root ganglion. And as you can see here, the denervation worked. Like TRPV1 goes down. This is the NAV 1.8 gene, and we don't affect the tyrosine hydroxylase expression. So it worked, and when you look at the genes of the thermogenic program, most of them goes down, and the animal cannot also resist to cold. We did this in females as well, and you have preservation of tyrosine hydroxylase, so sympathetic fibers are fine, and TRPV1 goes down, and NAV 1.8 goes down, so basically it really denervated, at least partially, and this was enough to deplete, to reduce the expression of genes of the thermogenic program. There is an interesting compensation of creatine kinase B, which is tyrosine hydroxylase expression. Somehow in females, they try to compensate in other ways here. But they can't compensate. Their body temperature drops even more when placed in cold, which means that their thermogenic fat is really affected. I didn't bring here for the sake of the time, but the morphology resembles those that I showed to you. Everything reproduce pretty well. And here is the expression of the tyrosine hydroxylase, just to show you that at least the innervation, the sympathetic innervation is preserved. In terms of expression, at least the integrity of the innervation, I would say it is fine. It doesn't seem that this sensorial, nociceptive sensory innervation is dealing with the sympathetic tone. However, we still need to dose norepinephrine with my friend here, Luis Navegantes. But here is what we really want to see is that if these manipulations that we did affect the neuropeptides, right? And indeed, when you give RTX, you have a very important reduction in CGRP. And when we use the IDTR, the diphtheria toxin tool, we also can see the reduction in CGRP. And the same thing we can see for the NKA. So basically, it seems that at least with the tools we have now, we can say that sympathetic tone seems to be fine. But on the other hand, the neuropeptides are reduced. So perhaps this innervation directly regulates the brown fat identity and function, plasticity, and not necessarily through a loop with sympathetic fibers like is done with the through piezo2 positive neurons. Okay. So this is code it's from an RNA seq from Yuhua Tseng lab that they kindly provided to us. So basically, they put mice in thermal neutrality, room temperature, and cold for 7 days. This is black 6 mice, and they did like a book RNA seq. And as you can see here, these are the two subunits of this receptor for CGRP. When you go to lower temperature, CGRP actually reduce. Okay, there are some evidence also that CGRP CGRP itself also reduce in cold. On the other hand, the receptor for NKA increase in cold. So it seems here. We are studying this yet, but it seems that they switch their roles. Like in room temperature and thermal neutrality, perhaps CGRP may be playing a role there, while the expression of NKA receptor is very low in room temperature, or at least in thermal neutrality, and and participates more in cold. Okay. When we look at receptor TACR2 expression in a single cell database, we can see here that is especially expressed in cold in adipocytes. This is adipocytes, and TACR2 is very expressed in cold in 7 days, also increase in cold in 2 days. On the other hand, the TACR1 is almost not expressed in adipocytes. TACR1 is the receptor, is the gene that encodes the NKR1, which is the receptor for substance P. Okay, and when you look at what happens in cold with the NKA levels, as you can see, NKA increase very much in cold, while CGRP reduce. So it seems that they switch roles, right, in room and temperature to cold. And when we treat adipocytes in culture, so here we move it to an in vitro setting because, okay, NKR2 is very well expressed, very induced in brown adipocytes. It seems that it has a role there. And let's see what happens if we treat adipocytes in culture with NKA. So NKA in 48 hours, in 24 hours, this is a representative of 24 hours, increase the genes of thermogenic program, and especially creatine kinase B. This is in mature adipocytes. When you look at the protein level, we also can see CP1 and creatine kinase B are higher. And also for you to understand the mechanism of action of this NKA, NKA can also hit a little bit NK1R and NK3R, but it's especially more active in NK2R. And it triggers a GQ signaling, so it triggers calcium dependent signaling. And in a recent paper from the Lawrence Casa group, they just showed that norepinephrine, while norepinephrine triggers GS protein-dependent signaling through cyclic AMP in beta-3 dependent way, alpha-1 receptor can also mediate some of the norepinephrine activities, especially through GQ and through the activity of CKB, creatine kinase B. So creatine kinase B seems to depend on GQ signaling, okay? So here our hypothesis is that while the sympathetic nerves can release norepinephrine to hit a GS protein-dependent signaling, the GQ signaling that boosts this norepinephrine thermogenesis can be elicited by NKA through these nociceptive neurons. So to prove that, we came back to the Brahma adipocyte setting, and we treated them with NKA alone. So this is acute. So NKA acute alone doesn't make any difference in the cells, just some effects are more chronic, but acute it doesn't do anything, as you can see here. But when you do a beta-3 agonist, it increases the expression of these genes of the thermogenic program, and when you put them together, you have a potentiation of the effect in terms of UCP1, PGC1 alpha, and also CKB expression, and TNAP a little bit too. And here is NK with forced choline is even more evident when you directly activate adenylyl cyclase, and together with NKA, you also potentiate this effect in the expression, but also you have the same when you look at it functionally in the seahorse setting. So as you can see, when you put NKA at 500 nanomolar or 1 micromolar together with CL, it potentiates the CL effect. So we have a synergistic effect here. And to see if this is GQ dependent, we use it first, we use this GR that is a NKA blocker. So here is the effect without the blocker, and then when we add this antagonist, we eliminate the effects of the NKA. And when we use an inhibitor of the, this is PGC1 alpha, we do the same. And when we block the GQ protein with GQ inhibitor, we also block this. So basically NKA is doing this through GQ signaling. This is in vivo. We also treated mouse with NKA in vivo, and we can see that NKA in vivo also increased thermogenic program. So it has a role in vitro and in vivo. So this is our working model. We think this NaV1.8 positive nociceptive fibers, they can regulate the brown adipose tissue function and the plasticity of the tissue. And one of the things that we can prove here is that NKA is sort of a neuropeptide, but very active neuropeptide that activate directly the brown adipocytes. And this happens through TACR2, GQ dependent signaling that helps to boost the norepinephrine dependent GS signaling. So this is my group. This work was done by João, as I said in the beginning. This is the fundings and my collaborators, Thiago Cunha, Yu Huatseng, and Isaac Xiu. So yeah, thank you.
We still have some time for question.
Thank you. Luis, beautiful work, very, very interesting. How do what do you think is activating these neurons? What they are connected to in the brain, or how they got activated? Do you have an idea?
At least in the subcutaneous fat, where there was this study with single cell sequencing of these fibers. These fibers express in those nerve endings in the tissue, they express leptin receptors, fatty acid receptors, or so basically it can be triggered by fatty acids from lipolysis, this kind of thing. So there are metabolic stimuli. These nerves are they are not part of flight or fight system, but it triggers fight or flight system, right? When you get hurt and this kind of thing. And I think the thermogenesis kind of do the same with this, but in the opposite of the mechanosensitive fibers that negatively regulate, it probably, we think this is triggered and when you need the whole machinery working, is to make a positive feedback, like to really boost thermogenesis, and without having an over-activation of sympathetic system, could be something like that. And yeah, but it's very complex. Like in thermal neutrality, or in room temperature, I wouldn't say it's not active. I think we are actually very interested in CGRP. Not in adipocytes. It seems that in adipocytes it's kind of confused, the phenotype. EASE is actually working more with that. We are more interested in what is in the CGRP in the vessels. RAMP1 is super expressed in the mural cells. So we are testing this hypothesis that also this can perhaps recruit the novel surge of brown adipocytes there. So basically, we believe in this balance between CGRP and NKA in different moments.
Nice talking, Luis. It's my turn. We have cut the nerves for a long time just to study the function of the sympathetic nervous system and the brown adipose tissue. So my question is, when you do that, when you cut the nerve, you take out the sensory innervation too? Because if yes, no, so we have to rethink what we are considering,
You mean, when I cut, I do denervate, but we are denervating especially just in brown fat, not in the endings.
Yeah, but this innervation come together with the sympathetic ones, right? They come together to the brown adipose tissue, right? The norepinephrine and the CGRP or whatever, and they come together to the tissue. When you cut the nerve, just to study the norepinephrine effects, you are cutting the sensory innervation too. So my question is, for a long time we assumed that we are seeing just the effect of norepinephrine, and it's not like this.
Yeah, this is what Lee proposed in the subcutaneous fat. He found that in the subcutaneous fat, 40% of the tyrosine hydroxylase positive fibers were NAV 1.8 positive too in the subcutaneous. So that is pretty much what you are saying, like they have an overlap. But what we found here is that these ones coming from DRG, dorsal root ganglia, are different. Basically, there is no overlapping with tyrosine hydroxylase in the ones coming from DRG. We have another study going on focused in the ones coming from the stellate ganglia. And there, where the major tyrosine hydroxylase ones are coming, there is a subset of tyrosine hydroxylase positive ones coming from there that are NAV 1.8 positive, a subset, not all of them. And this one is the one that wraps the vascular smooth muscle, while tyrosine hydroxylase is spread in the parenchyma. The NAV 1.8 not. They are more restricted to. So it's what you said is true for the subcutaneous, and is also true for this subset, but not for the whole. There are plenty of tyrosine hydroxylase fibers that are not positive. For TRPV1, there is no overlapping at all. I spoke with a our collaborator Ana Domingos, she did the single-cell RNA sequencing in tyrosine hydroxylase positive fibers. There is no TRPV1 there. But NAV 1.8, there is this subset, which we are studying in the lab. But it changed things, indeed, as you were saying. It changed how we understand this.
Thank you, Luis. We have to move
Thank you very
forward
much.
to stay on schedule. Okay, I will introduce Luis Navegantes. I hope I do not kill his last name. He's a physician and associated professor of physiology also at the Ribeiro Preto Medical School. And his research are focusing on metabolism, neuroendocrine regulation, skeletal muscle physiology, and particularly under condition of increased energy demand. Thank you, Luis.
Thank you. Good morning, everyone. Thanks a lot. Well, first of all, let me thank the organizers, especially Décio, for this nice invitation. It's a pleasure to come here, discuss our research with you, see my old friends, and it's a really nice to be here for my first time in this school. So today we're going to talk about the neuroregulation of autophagy. So in my presentation, I will give some focus on how the sympathetic nervous system can regulate this process in the liver, so shed light into the potential implications for health and disease. Okay? So you are pass? It's good? Okay. Now yes. So autophagy, as itself, it means self-eating, and it's a very important process just for maintaining cellular physiology by blocking damaged organelles or by providing energetic substrates. Okay, so that is the function for autophagy. And it depends on the autophagosomes. So autophagosomes, it's a double membrane structure that engulf and deliver cellular debris for degradation by lysosome. So it's a very important structure that is formed by a complex interplay of different proteins. One of these proteins is LC31. So LC31, normally, it's in the cytosol, in the type 1 form. But when you stimulate autophagy, it turns on LC32 in a bound membrane way. So this conversion from LC31 to LC32 is a reliable marker of autophagic flux. Okay, so the discovery of autophagy was very, very important, and let's remind that the importance of this discovery, no, it's not working. What? Okay, so, okay, agora foi, yes. Now, this importance, let's remind that it's awarded the Nobel Prize in Physiology or Medicine in 2016 to Yoshinori Ozumi. Okay, so the autophagy, the effects in autophagy are linked to several diseases like cancer, neurodegenerative disease like Parkinson, Alzheimer's, and metabolic disease like diabetes, obesity, and MAFLD. So for instance, we know that impaired autophagy may contribute to increase the hepatic steatosis. Because this disrupts the lipid balance and leading to a fat accumulation in the liver. So because autophagy affects metabolic disease, our lab has no, it's not working. Okay, so it's for the old way, you ask, so I can move this. So as I was saying, our lab, the interest of our lab is to investigate, the factors that control autophagy. So hormonal signals, the nutritional availability, and the central nervous system. That's the interest of our lab. The central nervous system controls autophagy through the sympathetic nervous system. So this system, as you know, consists of two main components. The adrenal medulla that release adrenaline directly into bloodstream, and the sympathetic nervous system or sympathetic nerve ends that release norepinephrine into different organs by regulating blood flow and metabolism, as Lydia said. Okay, so we know that for a long time that the liver receives a sympathetic innervation. We know that, but the microcircuitry of this innervation within the liver parenchyma is not well understood. So we collaborate with Dr. Ana Domingos at Oxford University and the Crick Institute just to understand this sympathetic innervation in the liver with more details. And for this, we use a technique called 3-disco. 3-disco is a method. Okay, let's see now. Okay, I will move that on the other side. Okay. Okay. But so, as I was saying, 3-disco is a tissue clearing technique that renders organ transparent. Okay, so you can see here that in this way you can see more easily the localization and how the innervation is with more details and importantly in 3 dimensions. You can see this with more detail. So let's see if you can. Okay. Pode colocar isso. Ok, vamos ver. Okay, let's see. This is the technique. That's okay. That's fine. To clear the tissue, you use different protocols until you get the clearing. And here you see. No, I think it's not working out. Okay, this is the old way. I think so. You can go. So okay, that's okay. So you can see here the background. Okay, in green, it's the background. Here you see after the tyrosine hydroxylase label. Okay, with this label, we can just reconstruct the sympathetic fibers, and you can get different parameters like area, volume, and so on, and you can quantify these fibers, okay? And in this movie here, okay, I think it's working, okay, so great. In this movie, you can see a liver from a control mice, okay, compared to a mice who was sympathectomized during the neonatal life. So you can see here that the sympathetic innervation, it has a large number of fibers, okay, and from these fibers, we have some accessory ones that are smaller, okay, but very importantly is that this kind of innervation, although they surround the blood vessel, it doesn't get deeply the liver parenchyma. In other words, it doesn't make direct contact with the hepatocytes. Okay? Okay, so let's go back. And, okay, good thing. That's okay. Yeah. Okay, that's fine. It's okay. Okay, move now. Okay, move now. Okay. Okay, that's okay. Okay. Okay, that's fine now. No, it's not. Ok. Onde que eu mudo agora? Vamos ver isso aqui. Okay, that's okay. Okay, just now, no, you can go if you want. You can come back, that's fine, okay. So here you can see that the same sympathetic network has been described in human livers. You can see here. So this paper come out just in the same moment that we are published our data in mice. And what is very interesting is that these fibers, these sympathetic fibers, they degenerate, okay, during the severity of human steatosis. Okay, so implicating this sympathetic innervation in the pathogenesis of this disease. So considering that autophagy is also impaired in the steatosis, we hypothesize that the activation of noradrenergic sympathetic fibers in the liver may lead to lipophagy during stress conditions, physiological stress conditions, okay? Providing substrates for energy production and help maintain metabolic balance. So just to prove this hypothesis, we use a very simple protocol, very, very simple protocol. We use sympathectomized mice treated with 6-hydroxydopamine, as you said before, and the control ones, and we expose these animals for a cold, okay, just for a few hours, during 3 and 6 hours. We choose this stimulus because it's very well known that colds increase the norepinephrine release from sympathetic fibers, but it doesn't affect the epinephrine secretion. Okay? We have to consider, of course, there is a lot of hormonal change during this situation. For example, the corticosterone, the glucocorticoid inhibitors, it goes up. Glucagon goes up. Insulin goes down. Okay, so there is a lot of hormonal change that that come together with sympathetic activation just to provide substrates during this situation, okay? But anyway, we will focus on the sympathetic activity during this situation. Okay, so the last thing we did was to look at the autophagic flux. And to do this kind of experiment, you treat the animals with leopeptin. So you do this because leopeptin is a lysosomal blocker that, okay, so that allow us to see the accumulation of autophagosomes. Okay, so here you can see that we look after we did that. You expose the animals, and you can see the conversion of the LC31 in LC32 here in the code during these 6 hours, and you see that this conversion increased, and it was blunted in the 6-hydroxydopamine animals. So suggesting that the parasympathetic innervation stimulates autophagy. In parallel, we see that the code also increased the autophagy-related genes, and this effect is also attenuated in sympathectomized mice. Using intravitreal microscope, we use LC3GF3 mice, and we image the livers of these animals in a real time. And after one single injection of norepinephrine, we saw that it increased the accumulation of autophagic puncta in the liver, suggesting autophagic stimulation. So this single protocol also increased the autophagic flux, as you can see here. So reinforce the notion that the norepinephrine indeed in vivo stimulates autophagy. Just to confirm these results, we move, of course, to the hepatocytes. And after incubating hepatocytes in the presence of catecholamines and also glucagon as a positive control, we saw the same. You see here that the norepinephrine and the catecholamines and, of course, glucagon, it's very well described, they increase the autophagy in hepatocytes. So it's clear with this data that the sympathetic nervous system seems to stimulate autophagy. But okay, what kind of autophagy? Mitophagy or lipophagy? So let's start with mitophagy. So here you can see that no marker of autophagy was regulated during cold exposure. You can see key markers like OPA1, MTF2, parking, whatever. It doesn't change during cold, and it doesn't saying that it's not controlled by the sympathetic nervous system. In addition, the number of mitochondria, the content of mitochondria doesn't move as well as estimated by citrate synthase activity and the DOX4 levels. So it doesn't seem that the sympathetic nervous system activates mitophagy. So we move to lipophagy, that, as you know, it's the selective autophagic degradation of lipid droplets. This process, okay, so it releases from these lipids substrates like glycerol and fatty acid that can be used as excellent substrates during energy demanding situations like cold and the other situations. Okay, so but before investigate lipophagy, it was important to see how the lipids were in the liver from cold-exposed mice, just to make sure about the substrate availability during this stress situation. So for this, we use the oil red staining that, as you know, it's a dye that binds to neutral lipids. And here you see that the cold dramatically increase in a few hours these lipids in the liver. So it was really interesting, but we investigated the underlying mechanisms behind this. Okay, so we tested, we use lipogenesis by tritium, and we measure the FFA as an indirect indicator, right? So indirect marker of lipolysis. So we saw that the fatty acid synthesis was not increased by cold, indeed it was decreased. So it could not explain this fat buildup in the liver. But the FFA, as expected, was high, indicating that lipolysis probably was high, and the increasing the fatty acid flux to the liver and the fat accumulation. Okay? Importantly, it's not modulated by the sympathetic activity. So let me show a recent experiment ongoing in my lab with a student, a graduated student, performed in adrenalactomized mice. What Gustavo did was just to take out the whole adrenal, so preventing the increase of corticosterone during this stress. And what you see here is that adrenalectomy blunts the fat buildup, okay? So demonstrating that cold release corticosterone increase the lipid content in liver. So it's corticosterone, not the sympathetic activity. And in agreement with the notion, you see here that adrenalectomy, okay, so just blunted the FFA increase, suggesting that the corticosteroid is increasing lipolysis, which increasing the fatty acid flux to the liver and making so the liver accumulates all this amount of fat. So, but it's important to reinforce that it's not regulated by the peripheral nervous system. Okay, so let's move it to lipophagy again, and to investigate lipophagy. Now we use the LC3 BODIP method. This is a very nice method because it allows us to see the colocalization of autophagosomes with in the lipid droplets, exactly in the lipid droplets, okay, colocalized with LC3. So you can see here a good colocalization during 3 hours of cold exposure of LC3 with these lipid droplets, indicating lipophagy. Okay, so we see here so that the cold, the hepatic innervation seems to increase lipophagy. Okay, so we hypothesize that these substrates from lipophagy, like fatty acids, could be beta oxidized and producing ketone bodies. Indeed, as you can see here, the beta hydroxybutyrate, you know, it's a very well-known ketone body. It increases in cold exposed mice, okay? And interestingly, sympathectomy not only blocks this production of beta-hydroxybutyrate, but also block the fatty acid oxidation estimated by Ouroboros. Okay, so these results, they tell us that the sympathetic nervous system, so it stimulates lipophagy, okay, and the cytogenesis. But what's the relation of lipophagy with the beta-hydroxybutyrate? Okay, so here what we did was to block the autophagosome formation by treating mice with leopeptin, just to suppress the autophagy stimulated by codes. And what you saw was that that the sympathectomy attenuated the production of the cetone body, okay, and interestingly, sympathectomy increased the TAG levels in the liver. So, showing that if you block the liver's ability to mobilize lipids, so you accumulate, you produce more fat. Okay? Okay, so the next question we addressed was what signaling pathway mediates norepinephrine-induced autophagy or lipophagy. So we know that, as Leiria said, norepinephrine can stimulate different signaling pathways, but in the liver, the main adrenoceptor is alpha-1. So it's very well known. So in a recent paper, we demonstrated that the stimulation of norepinephrine in liver could increase the gluconeogenesis by activating a calcium signaling probably mediated by alpha-1 adrenoceptors. And here you see some markers of this calcium signaling. You see that cold exposure increased the PKC substrates, the CAN kinase 2, the levels of CAN kinase, and sympathectomy blocks these effects. Okay? In hepatocytes, you see that norepinephrine can increase the levels of CAN kinase 2, suggest again that a calcium signaling pathway was being activated by sympathetic nervous system in the liver. Okay, but how it could be related to autophagy? So this is our hypothesis that norepinephrine in hepatocytes, it's stimulating alpha adrenoceptors, activated GQ, and promoting the activated of phospholipase C. Okay, so increasing the IP3. IP3 can link to IP3 receptor and mobilize calcium in the cytosol. Increasing calcium in the cytosol, you can activate autophagy. We don't know exactly how, but we have a lot of evidence. For example, calcium can stimulate the CAN kinase 2 and stimulate CRAB, and CRAB could increase the autophagy-related genes. Another possibility is that calcium can induce autophagy through AMPK or whatever. We don't know exactly, but what we did was just to trying to understand the whole of IP3 and the CAN kinase 2 for autophagy. And these experiments were recently did by a PhD student at Schumann's lab. And what we did was just to use this double GAL-NAC-azole. It's a kind of it's a way to knock down the protein you want in the liver, right? So we use three kinds of knockdown mice. IP3R1, kinase 2, or the combination of these two proteins. And what we recently saw is that the autophagy, as you can see here, is blocked in these animals, in these knockdown animals here, suggesting that these proteins are involved in the autophagy mobilized by sympathetic nervous system. Okay? Okay, but it's very preliminary, but so far we can just make preliminary conclusions. The first is that cold exposure activates hepatic autophagy, particularly lipophagy, via sympathetic noradrenergic innervation that triggers calcium-mediated signaling through the IP3R1 kinase 2 pathway. This response promotes lipid droplets breakdown and cytogenesis, while disruption of sympathetic input impairs this process, leading to hepatic fat or contributing to fat accumulation. However, the accumulation of fat in the liver of animals acutely exposed to cold is most likely due to the high influx of fatty acids resulting from lipolysis stimulated by glucocorticoid, suggesting hormonal interactions between corticoids and the sympathetic nervous system in this physiological situation. So here my acknowledgements for all my group, my colleague, Dr. Isis do Carmo Ketelut, Dr. Millerini, our mentor, as William is here. Here are the group in Ribeirão Preto, the technicians, and especially my thank to Henrique Morgan, who did most of these experiments, the fundings, and of course, you for your attention. Thank you so much.
Thank you, Luis. It was a terrific talk, even if we got some problem with the video. Thank you so much. Time for question, if you have some.
Very nice talk, Luis. You basically saw that under physiological condition, sympathetic stimuli does not induce mitophagy. But do you think that it can regulate mitophagy under pathological condition, let's say under Western diet or something that it will like dramatically affect the liver? Do you guys have any plan to do something like that? Thank you.
I have no evidence about this in the liver, but we have studied in other tissues. And I don't want to complicate you, but in other tissues like skeletal muscle and the brown deposit tissue, it's the opposite. Okay, so it's completely the opposite. So perhaps because the sympathetic nervous system seems to protect mitochondria, for example, in the bat, right? And it's very clear if you denervate bats, the mitochondria goes down. Okay, so we demonstrated recently and in a paper that the mechanism underlying this is autophagy. You have autophagy. Okay, so you take out the innervation, have autophagy, you see the catepsin enzyme activity increase dramatically, the LC3, the puncta NOA. So suggesting that the stonus in the bat, for example, protect mitochondria. But in the liver, I have no evidence of that.
A very nice talk. Your leupeptin experiments in vivo are very nice.
Thank you.
I just have a suggestion and a comment.
Right.
I think it would be nice to measure P62 as well, if you can, in the mouse P62, it's a autophagy substrate, because sometimes what happens is, although we see changes in autophagism and autophagic vesicles, you also want to ensure that lysosome is fine and the substrate clearance is happening. So yes, the leupeptin is beautiful experiment. Not everybody can do it in vivo, so it's great. But if you couple that with P62, you have the tissues. You can just do the Western, would be fantastic. And I can also suggest some good antibodies for mouse tissues.
Yeah.
And regarding calcium, it has been a very, complex scenario in terms of autophagy. There has been a large number of papers saying that if you use thapsigargin, for example, which stimulates calcium, it massively blocks autophagy. That means it prevents autophagosome lysosome fusion. Likewise, the calcium coming from extracellular via the L-type calcium channel, if you use a compound called BK, which is an opener of calcium channel, it can also impair autophagy. However, localized calcium signal can stimulate or affect, induce autophagy. So maybe you can, refine your conclusion saying that it could be a localized calcium signal and not massive efflux from intracellular stores, and it also could be a tissue-specific scenario which is happening in your cell type and not universal. So this will thank you.
No, yeah, it was excellent suggestion. Yes, P62 levels, yes, it's missing here. We have looked at, I don't know why we didn't show here, but it's very protein, very essential just to conclude anything. Yes, your observation is really nice. And we'd love just to see the calcium localization. But I have to say that I'm really afraid of this, because I'm afraid of calcium signaling, okay? Because it's really complex. So that perhaps this workshop, helps me just to overcome this fear. Huh?
Other question? Well, it's time for the coffee break that you are waiting for. I just ask you to be back at 10:20 to be on schedule.
Sorry for the...
No, that's not your fault.